Welcome to our 9th online series. So this is the 2024 spring series. We're focusing on aging. I'm Jeffrey Guy, founder and chairman of the foundation. And for those of you who are new to us, we are promoting an interest and further research into quantum effects in biology, how they may affect health, disease, and give us opportunities for therapeutics and diagnostics for the future. Now, in the previous symposium, we've covered everything from some very fundamental quantum mechanics up to practical applications of these new thoughts in therapeutics. And in the autumn last year, our series focused on space health, and this is a very nice segue to the series for spring, which is focusing on aging. Now, we're going to look at the basic principles and current theories on aging, and our speakers will be Ken Raj, Wayne Fresh, Nick Lane, Alistair Nunn. And today we're going to welcome Pedro Magalhaes from University of Birmingham to speak today, and Alistair will introduce Pedro. So Alistair, who I think you all know by now, is our Director of Science for the Guy Foundation. He's also visiting professor, University of Westminster. Alistair is going to introduce our speaker today. And without any further ado, Alistair, I'm going to hand over to you.
Okay, yes, thank you very much, Jeffrey. Just going to, I think before we introduce me, I think everybody knows who I am. Before we get to the main event with Pedro, I just thought I'd put together a few thoughts and then introduce them. It won't take very long, just to get us thinking perhaps slightly differently and where we've come from. And so why as a foundation who are interested in quantum biology thought about aging? And I think the truth is, of course, aging is really fundamental to everything when we start thinking about disease. And we've long thought actually that perhaps approaching biology from the quantum aspect might, in some levels, give us a better insight. And certainly, ultimately, we all know as we get older, we tend to get more disease. And the situation at the moment is really, despite a lot of research as a species, although we've got some understanding of it, we're still not really any closer to slowing down the aging process as a whole. And certainly this is demonstrated because the modern lifestyle seems to be accelerating the aging process a little bit. And this is something we've studied up at Westminster. But we're also very interested, again, as to how we might apply other models of aging. And one of the models which appears to be coming very much to the fore is what happens to astronauts when they go into space. And this is this idea of the accelerated aging phenotype. Obviously, we're also interested in mitochondria. I won't go too much into detail. But this is kind of a question that Jimmy and I have often asked ourselves is actually what is health? We often talk about disease, but what's it like to live in optimal health? And indeed, this is the reason that Jimmy set up his Centre for Research on Optimal Health at Westminster. And this is like a very simple diagram. I know there's a lot on it, but fundamentally, how long do you think humans would live if they live in perfect health? And if you look at the data, it suggests that if everybody lived perfect health for all their lives and everything went right, many of us would get to, close to 100. Obviously, there's genetics involved, which I'm sure Pedro will tell us. But one of the key things that we all seem to understand is that we actually, if you live in health, you tend to have a good, healthy lifespan too. You tend to live quite a long time without any serious problems. But what happens in the modern world, we seem to see that actually life expectancy is nearer to 80 to 85. And folk often develop conditions we might call disease for a bit longer and a bit earlier in life. And if we live a very poor lifestyle, and by this I mean if we sit around all day, eat fried burgers and don't eat any fruit, and just generally live a bad lifestyle, we actually get our life expectancy even drops even further. And we see things like metabolic syndrome, diabetes, cardiovascular disease, you name it, it's all there. And this is called morbidity expansion. And so the question kind of is, how do we go from here back to here again? And is it possible to go to here? And one of the things that seems to be popping out of all of this is this concept of metabolic flexibility. And this is the ability of the system homeostatically to adjust to stress. And this comes back and it has an important role in the aging process. And one of the interesting factors, and this goes back to this idea of the modern lifestyle, is there was a paper published a couple of years ago. And I don't know how true this is, but the data seems to suggest that certainly in North America, less than 7% of people may actually be living in good cardiovascular health, which kind of tends as, you know, we're not doing very well as a species at the moment. And the takeaway message really from this is it seems that by and large, lifespan or life expectancy can be modulated quite a large degree by the environment and how healthy we live. You know, what do we mean by, well, aging? And this is something again Pedro will cover, and this is just another way of showing the aging thing. And certainly we're now seeing with all these new markers and techniques, we can see that people's chronological age isn't always their biological age. And certainly an accelerated aging phenotype seems to be that they accelerate the aging faster, and they seem to be biologically older, and vice versa. And certainly there are all sorts of markers now that everybody's picking up in terms of the aging process, from genomic instability to things like epigenetics, failing ability to digest our own intracellular organs called autophagy, certainly mitochondria. Cellular senescence is a big one. Of course, cells that stop dividing, they sit around and become inflamed. Our stem cells don't work. And we also see things like declining cognitive function and things like dysbiosis, which is caused by microbiota. But two things which I think are really quite interesting is if we look back through time and look at what is the oldest markers of aging, and by this I mean going back through species and phylogenetically, it seems that proteostasis, and this is the ability to maintain your proteins in good shape, seems to be one of the earliest things as an indicator of aging. If you go back to very primitive organisms. And in a more modern world, what we see, and this is this idea that actually as we age, we seem to get more and more inflamed, and we tend to show more levels of oxidative stress. So in fact, I think most people probably kind of understand this. And so what do we mean by aging? Well, there's multiple kind of interpretations, but one of the ones is we seem to experience a gradual loss of the ability to adapt to stress. And as we get older, we become more and more susceptible to disease. And it seems to be associated with a gradual reduction in the ability to maintain homeostasis. And certainly when you look at different species, this seems to be a process of adapting to the environment. So basically, you can argue that there seem to be some species which breed very fast and don't live very long. And this seems to be adaptation to a certain environment, whereas some species live a very long time, and Pedro will talk about this. And they seem to be able to outwit the environment to some degree. And there are certain factors which we can see, you know, across species. Certainly large size is one of them, intelligence is another, and an ability to get away from predators, for instance, being able to fly. There are exceptions to this. The octopuses, for instance, are very smart, but they don't live very long. But most of the time, it does seem to be this is the case. And certainly there was old ideas about the rate of living theories, and this is the idea that you've only got a limited number of heartbeats, etc. And you know, the more you exercise, the more you work, the less long you live. Well, actually, this doesn't seem to be the case, because you can have animals of very similar sizes, like rats or mice, which only live 3 or 4 years, but you can have bats which live 30 years plus, and then naked mole rats, which also live a long time. So clearly, nature can modify the aging process. And so really, what is it? It seems to be about a molecular degeneration of structure. And from our perspective, of course, we're looking at this from slightly more from the quantum mechanical perspective. Of course, it seems to be about the ability to channel electrons safely. And you know, if the electrons go to the wrong place, we end up with oxidative stress and inflammation. And is this something, you know, relating to entropy? And of course, inflammaging, which I think certainly Pedro can talk about, is something which is something we're all really familiar with now. One of the things we find very interesting, and certainly, and I know Doug Brush finds this, it's an interesting concept, but this is the concept of hormesis. And this is the idea that actually biology can adapt to low levels of stress and become slightly stronger and more robust. And basically, what doesn't kill you makes you stronger. I think you've all heard the song, but it also seems to make you smarter and longer lived. And actually, if you think about it logically, it's obvious it's important in evolution. Without stress, we wouldn't have evolved to anything. So clearly, it's part of what biology is. And it also seems, for instance, the ability to resolve inflammation. And again, it's back to one of the factors we can see involved in this is a modulation of redox and again the flow of electrons. It's been called many things over the years, but one of the ones that most people understand is the concept of allostasis, which is basically stability through change. Basically, it's a biphasic response to stress. And we've thought about this for a long, long time, and this is a very early attempt at one of our papers. But we suggested that, you know, when we look at modern disease and the incidence of metabolic syndrome and diabetes, and largely it seems to be due, certainly in modern lifestyle, is not exercising enough and eating too much. I know this is very simplistic, but actually you could argue that it's a lack of hormesis. But one of the key things to think about here is that we can argue that that life displays something like a flight envelope, or we call it adaptive metabolic envelope. And without stress, we've become canalized to quite a narrow area, and any stress outside of that causes a lot of problems. But with a bit of stress, we seem to expand our envelopes and be a little bit more robust. And certainly we see evidence of this now, certainly with things like COVID-19. And this was something which a lot of people, including us, jumped on very quickly. And so, well, which populations of people are going to be affected the most? And certainly it seems that those with comorbidities, and certainly those with a metabolic syndrome, and we would argue have less mitochondrial function or lower mitochondrial function and metabolic inflexibility seem to be far more susceptible. And this does seem to be turning out to be the case. But of course, there's an even bigger question here: is why do we age at all? Why aren't we immortal? What is it about nature and life that means we don't live forever? This goes right back to the heart of the role of evolution and the beginnings of life. Is it genes or metabolism first? If it's genes first, well, were genes involved? Was metabolism important? The role of natural selection. And we would argue that is thermodynamics and quantum mechanics important. And certainly it seems, if you think about the electrical analogy, that life is electrical because it's about movement of electrons, eventually, the circuitry doesn't seem to be perfect, and it doesn't repair itself properly. And is this part of how life adapts to new environments? And one way of viewing this, and this is just kind of thinking slightly from the side here, but one way of viewing this, we go back to ideas of how life started, and one of the ways that life may have started is in this alkaline thermal vent. There are other theories available, and I say that, but this is the one we quite like this one because it kind of reflects potentially where chemiosmosis came from. This is the proton gradient, which is generated by the electron flow, and this is Peter Mitchell who came up with it. But this chap here, Rick Chance, he said a long, long time ago and really championed the idea that quantum tunneling was important. But of course, folk were talking about this 100 years ago, and this fundamentally begins to suggest that if we think about the electrical flow, is aging is something to do with the degradation of this system involved in aging, which I think is quite an interesting concept. And certainly, as we know, this one of the aging components we know is mitochondrial dysfunction. It isn't the only one, but we know the mitochondria came along quite a long time ago and key in eukaryotic life. So really, it's quite possible that when we start to think about aging, maybe we need to think a little bit more broadly about other aspects that we can apply to it. This includes both thermodynamics and quantum mechanics. I won't go into what this diagram means, but I think most people see it. But basically, we often think about biochemistry and just the movement of molecules. But when we start to think about this, we also have to start to think about what application of quantum mechanics might say about the role of fields, for instance. Every time an electron moves or a proton moves, it generates an electric field. We may have to think about this in how this all comes together in life. So when you think about this series of talks, which we're going to have this in the spring season, think about this slightly from a different perspective and see how this might mesh with what we're going to hear in the next 5 talks. So any more ado, I'd like to introduce you to Pedro. He is Professor of Molecular Biogerontology at the Institute of Inflammation and Aging at the University of Birmingham, and is looking for ways to try and find how we can understand better the aging process to help improve disease, reduce disease, in particular from the epigenetic and comparative genomics perspective. He has over 16,000 citations with an H index of 66, which is very good. Over 113 published articles in the field of aging. I'm sorry, the source is Google Scholar, so I apologize, Pedro, if that's not quite true. But I've, having read many of his papers now, one of my favorites was the one with the dinosaurs. That the fact that this echo of how we evolved millions of years ago and how we were, how mammals came into existence has actually dictated how long we live now. So today he's going to talk about genes regulating aging and the quest for immortality. So without further ado, I'll hand you over to Pedro.
Thank you very much for the great and comprehensive introduction, and which touched on some of the topics that I will mention today. So my goal for today is to provide an overview of the biology of aging. I'll mention some of the works we've done, but mention a lot of what we know and don't know in the field as a whole. So I'll start with quite a long introduction, actually, because I know this is the first talk in the series. So I'll start with quite a long introduction about the biology of aging, genetics of aging, what we know, what we don't know. So it doesn't go, my talk, I will mention some of the works we've done, but I won't go into a lot of methodological detail on that. It's more of a high-level overview of the field, and then I will touch upon some specific projects that we've been involved in. So actually, so I mean, I suppose aging is, one of the advantages working on aging. They're very familiar with it, and as I'm sure you're aware, since the beginning of time, that many, particularly men, have sought the eternal youth or sought to rejuvenate themselves. Qin Shi Huang, who was a Chinese emperor, he built a terracotta army. So he tried to become immortal, tried to become young again by taking these pills. But unfortunately for him, the main component was mercury, and so ironically, he died of mercury poisoning. But there's been many others since then, and I think the first scientific approach to rejuvenation was a Russian scientist from St. Petersburg called Alexander Bogdanov. And he tried to achieve rejuvenation with blood transfusion. So there was a scientific basis to it. The idea was that if you get blood from young individuals, then this will have a rejuvenating effect, which we actually now know that well, to some degree, there are functional benefits to it. Unfortunately for him at the time, he wasn't aware of blood types, incompatibilities, and so on. And so he also sadly and ironically died after a blood transfusion from a young student. But it was the first scientific approach to rejuvenation. And the point is that, nobody wants to grow old and die. We all would like to be younger for longer. And that's, I think, intrinsic to the human existence. But now for the first time, we have, I hope to convince you, at least a way of maybe not become young again, but to slow down aging. So what is aging? Well, I'm sure you all can recognize the process of aging, as you can see in this Czech Centenarians. I tend to have quite a broad definition of aging. I mean, obviously, aging involves many changes: physiological, functional, molecular, cellular changes, pathological changes at different biological levels. I tend to have a quite broad definition. I've been using this for many years, which is aging. I tend to define as a progressive deterioration of physiological function, accompanied by an increase in vulnerability and mortality with age, which, yes, it's broad, and it encompasses many facets of aging, including physiological degeneration, increased vulnerability, and increased mortality of aging. With age. One of the hallmarks of aging is that once you reach about age 30, your chance of dying doubles roughly every 8 years. And that's very consistent across populations. So you see it in the UK, in the US, but in African third world countries, you see the same pattern. So that's very consistent hallmark of aging. So that's, I would say, again, a broad definition of aging, and I'll dig into a bit more detail later on. And as Alistair mentioned already, we have a number of diseases that are influenced or whose incidence increases dramatically with age. This is data from COVID-19 that I'm sure you're familiar with. This is mortality from COVID-19 with age, which, as you can see, is fairly low until you reach about age 50, and then it increases exponentially. So something changes in our bodies, presumably in our immune system. There's immunosenescence. At some level, we don't quite understand, but that means that someone who's older, an older individual getting COVID-19, has a much, much, much higher chance of dying than someone who is younger. Again, it's an exponential increase. And you see it for many other diseases. The major killers of modern societies, this is data from the US, will be similar for the UK, are age-related diseases, as I'm sure you're aware, like heart disease, cancer, neurodegenerative diseases, type 2 diabetes, and infectious diseases as well. Things like pneumonia and, the common flu are a significant cause of death in elderly immunocompromised individuals. So it's a major medical challenge, aging. As Alistair already mentioned, if we could do something about it, if we could slow down aging, even if slightly, this would have massive medical, social, economic benefits as well. The problem is that we don't know why we age precisely. We don't know the specific, precise molecular cellular mechanisms driving aging. There's a number of hypotheses. I'll come back in a little bit that Alistair mentioned as well. Oxidative stress, DNA damage, telomeres, senescent cells. There's many hypotheses, but they are just that. They're just hypotheses. We don't know for sure why human beings age. I would say we don't know even very well why human beings age. We don't know. We have hypotheses that we haven't really tested properly. And I'll come back to this in a few minutes. Now, There are reasons to be optimistic, however, that we can do something about aging. On one hand, there are species that appear not to age at all. Doug Finch called species exhibiting negligible senescence. And as you can see, these are complex animals, vertebrates. We're not talking about little amoebas. We're talking about, some fishes like rockfishes from the genus Sebastes, some turtles and tortoises. This is a blind salamander that lives in caves in France called the horn. So these species, when you study them, for example, they don't exhibit the exponential increase in mortality that you see in human beings and all mammals. They don't exhibit, at least not obvious, physiological degeneration. Of course, maybe they age after 500 years. I mean, it's hard to get PhD students focused on a 4-year project. So, we don't know for sure. Maybe they will age after a very, very long time. But the point is that at the very least, they age much, much slower than human beings. So as Alistair touched upon, clearly evolution has figured it out to some degree. That there is a way. It's not like there's a physical limit that say, that it's inevitable. Aging is inevitable. Well, perhaps, but at the very least, there are species that age much, much slower than human beings. And the other breakthrough in the field, which I'll focus a little more, is really the demonstration that we can manipulate aging in animal models. In particular, at the genetic level, there's also dietary and pharmacological interventions that I'll mention. I'll mention pharmacological interventions a little bit. I'm not going to talk today much about dietary manipulations of aging. But we know since the pioneering work of Tom Johnson and colleagues in the late 90s in Colorado, that individual genetic variants, so single genes, can have a significant impact on aging and longevity in animals. So these were experiments initially conducted in C. elegans. So essentially, you can tweak a single gene and significantly extend lifespan in these animal models. And Cynthia Kenyon, another pioneer in the topic, points out, in theory, what it shows is that maybe we can change a human gene and significantly increase the human lifespan and health span. So, this is quite a remarkable breakthrough in the field. So one of the things that I studied as a PhD student, I thought it was fascinating, that you can just change a gene and increase lifespan significantly in animals. So, back when I was a PhD student 20 years ago, I studied this collection of databases and tools called the Human Aging Genomic Resources because I was fascinated by it. I'm a computer geek. And so instead of keeping track of all these genes in an Excel spreadsheet, I thought I'll make a website to make it available to the public. And so one of the databases we have is called the GeneAge Database of Aging-Related Genes. And these are proven very popular websites and databases with thousands of visitors, hundreds of citations. But the point is that these reflect how much we've advanced in the field at the level of our understanding of the genetics of aging in model systems, what are called the gerontome. So this is from the latest version of our database. I would point out we now know of over 2,000 genes in model systems that. So we now know of over 2,000 genes in model systems when you change them. So when you activate them, when you knock them out, when you silence them, when you mutate it, some level of specific genetic manipulation that has a significant impact on aging or longevity. And it's over 2,000. So in worms alone, we know of nearly 900 genes. And the current record is a 10-fold life extension. So single gene, you tweak in worms, and these animals, which are like just microscopic roundworms that normally live 3 weeks, well, a few weeks, let's say, it depends on temperature as well. Instead of living a few weeks, they're living over 6 months. So that's quite remarkable. If we could do that to humans, we'd be living over 1,000 years. Now, of course, worms are, you could say, well, tiny microscopic, mostly post-mitotic animal, it's very simple. Well, even in mice, okay, the effects are not so big, but even in mice, you can knock out a single gene and extend lifespan by up to 50%. So okay, it's not as much as in worms, but it's quite remarkable. Again, if we could do that in humans, we'd be regularly living over 150 years. So particularly in mice, you can also accelerate aging. So you can create mutant animals that have a short lifespan, and they appear to be aging faster. it's obviously, but what does it mean to age faster? I mean, just because you have a short lifespan doesn't mean you're aging faster, right? I mean, maybe you just have some illness. Well, to give an example, these are two mice. I think this is a mutation in a gene called ERCC1, which is a cell cycle regulator. The animal on the right, as you can see, it's got, hair graying, a curvature of the spine. It has signs of premature aging that you don't see in its wild-type littermate. So, at least to some degree, or at least some aging phenotypes, you can accelerate in mice. And conversely, you can retard aging. So these are two mice of, again, the same age. The difference is that the animal in the bottom has a mutation in growth hormone signaling, which is why it's smaller. And so because it has this mutation, they're smaller animals, they're resistant to cancer, and they also live quite a long time. So this record about up to roughly 50% increase in lifespan is from disruption of the growth hormone signaling in mice, which extends lifespan. So what about human beings? Okay, obviously we can't make genetic modifications in human beings, but you can investigate individuals that have some of the same mutations that slow aging in mice. So this was a study from Walter Longo and Jaime Guevara-Aguirre. This is Dr. Guevara-Aguirre here, and he decided to study, as you can see, small people. So these are individuals that have a mutation in growth hormone receptor. So they're smaller, and if you knock out growth hormone receptor in mice, you have small mice. You have these actually very cute little dwarf mice, and they live longer in their cancer resistance. So what they found most of all was that individuals are very strongly protected from cancer. So what you see here is growth hormone receptor deficient individuals, and these are their relatives. And sample size not massive; it's about 30, but there were no individuals. None of these small people died of cancer. So that's quite interesting because the relatives, about 20-25% of them died of cancer, which is what you'd expect, which is kind of what you'd expect in the UK. And about 50% of individuals develop cancer in their lifetime, unless you have this mutation, in which case you have protection. Now, I should say these individuals, they don't live longer than normal. They have a normal lifespan. They have a high incidence of cardiovascular diseases. The way Walter explained this to me was that because they're smaller, their arteries are also smaller, and so they clog up with cholesterol smaller. So I actually don't remember seeing specific studies on it, but I think that's a reasonable explanation. They also have a high incidence of certain neurological conditions. Also, there could be behavioral factors there. But the point is that these individuals have the same lifespan of their relatives, but they're strongly protected against cancer. So this mutation that impacts on aging in model systems, in human beings, it protects against cancer, but doesn't have such a big lifespan effect as it does in mice. Now, what about, exceptional human longevity? I think Alistair was talking about, how long in optimum conditions human beings can live. Well, I think this amazing lady called Jeanne Calment from the Arles in the south of France is probably the best example. She lived to be 122, and this was more than 25 years ago. So it remains the record longevity for the human species. And it's quite interesting because she loved port wine from my hometown of Porto. She ate lots of chocolate, and she only stopped smoking later in life. When she was over 100, because she was afraid she would hurt herself with a cigarette because her eyesight was getting worse. So she really didn't have the lifestyle that you kind of associate with the longest human being on record. And she's not an only case. When you look at centenarians, some of them have very healthy lifestyles, others don't. It's not like these are the optimal health kind of individuals. A lot of them don't have very healthy lifestyles. It does tend to run in the family. So we do know there is a correlation. If your parents are centenarians, then there's a much higher probability that you're going to be a centenarian as well. And you can do studies of that. There's been done in twins, for example, at King's College. You can look at twins, and you can actually calculate heritability of longevity, which is about 25%. For comparison, the heritability of height, I think, is about 50%. So the heritability of longevity is not massive; it's moderately heritable. So that means that it has a moderate genetic component to it. However, it increases with age. So what that means is that if you want to be a healthy 70 or 80-year-old, it's mostly down to environment. It's mostly down to your diet, to lifestyle, and so on. But if you want to be a centenarian, it becomes more about genetics. So yes, it's about, if you want to be a centenarian, it's mostly about choosing your parents and grandparents well. Now, one challenge we still have in the field that I should highlight is that we don't know what genes are involved. You can say, yes, there are families in which longevity runs, but we don't know which genes are associated with the longevity, exceptional longevity of the families. So that's still a big mystery, I would say. I think the point is that aging is not just a sum of age-related diseases running individually. There are correlations between them. It is possible to manipulate the aging process as a whole. So, one thing I forgot to say is that, the long-lived mice, it's like the growth hormone receptor mice, little animals, they don't just live longer. They are living longer with good health. In other words, they still develop age-related diseases, of course, but they develop those later in life. So their health span is also increased, which is quite important because, obviously that's what we want. We don't want to live longer in a period of decrepitude. We want to live longer with good health and enjoy those extra years, right? So now, and in animal models, we can do this. Now, of course, we cannot manipulate genes in human beings, at least not yet. But perhaps manipulations by diet, by pharmacological interventions that retard aging could dramatically improve human health. If we could retard aging, even if slightly, if we could retard human aging, it would have a massive health, social, economic impact. Now, the other aspect that I would point out, it goes a bit more towards my work, is that, as I said, you can tweak a single gene and have an impact on aging. And that's remarkable because the human genome is massive. 3 billion base pairs, 20,000 protein-coding genes, God knows how many non-coding genes. And yet a very subtle change in the genome can have a significant impact on aging. You can have very subtle changes in animal models that extend lifespan or accelerate aging. So if we could decode the genome, if we could understand that, then that would really open the door for us to manipulate aging in a much more efficient and precise ways. So one of the things that, as I'm sure you're aware, there's this massive amount of data being generated from sequencing, from gene expression, from transcriptome. So really there's a shift in biology and biomedical sciences from generating data, which is a lot cheaper and easier and quicker now, to analyzing that data and interpreting and making sense of it. So I think there's been a shift in the way we do biomedical research, not just in aging, but in other fields as well, and disease-specific fields of analyzing, focusing on the data, and analyzing, processing, and making sense of the data as opposed to generating the data. And that brings us somewhat to what we do in our lab. So I run the Genomics of Aging and Rejuvenation Lab. My goal is really to understand the process of aging, genetic, cell, and molecular mechanisms of aging, and develop interventions towards it, with this understanding that, aging is a complex process. It involves multiple genes and pathways, and really we need to make sense of the genome and of biology as complex systems, not just of one gene. And we do different types of things. So our focus, of course, is on aging, particularly from trying to understand how the genome regulates mammalian aging. We do some work on dietary manipulations and pharmacological manipulations. I'll mention pharmacological manipulations a little later on. Evolutionary aspects, which I'll briefly mention as well. The relationship with diseases. We do some experiments in cells. I'm actually a cell biologist by training. We have some work on long-lived species, which I'll come back to in a little bit. And then trying to understand the process of aging from a theoretical perspective, and also a systems approaches, as understanding aging as a system, not just one process, but as a combination, which I'll come back through in a little bit. So the argument is that, okay, we know that biology is complicated, and although you can have simple processes, Mendelian diseases derive from mutations in a single gene, although could have then complex cascade down the line. The point is that to a lot of human phenotypes, like longevity, like aging, they don't derive from a single gene, they derive from the interactions of multiple genes with each other and with the environment. And so we need to understand how together the genes determine a given process or phenotype. So we need to understand their connections between what to understand the whole from its parts. So that's one of the fundamental concepts of our approaches and of our lab. And I'll show you a couple of examples. So, I mentioned about genome before. We know of over 2,000 genes associated with aging in model systems. But we can reduce the complexity. We can group them into common pathways. Depending on when a particular gene mutation increases or decreases lifespan, we can also classify genes as pro or anti-longevity. So that's what we've done some years back. And what we obtained was essentially a list of the major processes and pathways impacting on aging using this unbiased approach, just looking at manipulations in animal models. So for example, most of these would be pathways you'd expect to find, and they haven't changed much in recent years. But so for mice, for example, for longevity, you have cell cycle regulation, p53 signaling. For anti-longevity, you have growth hormone, insulin signaling, IGF-1. Because if you mutate growth hormone receptor, you get an extended lifespan. So those are anti-longevity pathways, which are quite well established and studied. For pro-longevity, you have hypoxia responses in flies, and anti-longevity, you have oxidative phosphorylation, PI3 key kinase, which sits below growth hormone IGF-1 pathway as well. And in worms, for pro-longevity, you have autophagy, which I think Anstar also briefly mentioned as well. And anti-longevity, you have oxidative phosphorylation and mTOR signaling. So nothing revolutionary. These are the major pathways you'd expect to find anyway. But these are, using this unbiased approach, some of the top pathways you find that are associated with pro or anti-longevity in different model systems. Now, the other thing, of course, is that because we have all of these manipulations, you can use them to test mechanisms of aging. As I said before, we don't fully understand mechanisms of aging, but we have a number of hypotheses. There's the idea that oxidative damage, for example, impacts on aging, epigenetics, protein aggregates, mitochondria, cell senescence, changes in stem cells, telomeres, and DNA damage. So there's a lot of hypotheses, a lot of mechanistics of aging, and we can test this. We can test them to some degree using human genetics. That's harder because, there's only, as I said, we don't really understand very well which genes are associated with longevity. But there has been some insights from it, and in particular in model systems, you can, take one of these pathways or take genes associated with oxidative stress and mutate them in mice and see what happens. So that's been done to some degree, although with largely subjective results. And one of the problems, I would say, is that because biology is complicated, we don't truly understand the relationships between different processes. So you can have, you can have very simple relationships where, like a Mendelian disease, where you have a mutation in a gene and it results in a phenotype. But biology is, in particular, it's, nonlinear, in multiple feedback loops and complex causal relationships. So just because you manipulate a particular pathway and it has a given output, let's say longevity, it doesn't mean it's a straight line. I would argue in aging, it's not going to be a straight line. And so understanding these relationships between causality, I would argue it's quite complicated. Even for simple processes. So I've just published this perspective in Nature Genetics that goes into more detail. But, just a demonstration that smoking causes lung cancer, that was not simple. That involved a lot of epidemiological evidence, studies in cells, studies in mice. It was quite a lot of work just to prove something we now consider obvious. And so to demonstrate that any mechanism has an impact on aging or is a driver of aging, I think that's particularly complicated because of this non-linear causal relationships in biology, and also because there is a lot of subjectivity to how you interpret, for example, is a short-lived mouse model accelerated aging or not. And so we're still at the stage where we don't well understand what are the driving mechanisms of aging, although some of them have more evidence than others. And I'll come back to it in a little bit. So the other kind of approach we take to make sense of all the data is to employ machine learning, AI, let's call it data-driven approaches to understand, to study genetics of aging and also manipulations of aging. And I'll give you a couple of examples. I mean, by and large, we use what's called supervised machine learning. So, a good analogy is if you think of, sometimes you see it on Facebook or websites to determine which is your Game of Thrones character. So the way those websites work is that they'll ask you a series of questions, are you male or female? Do you enjoy killing people? Yes or no? And depending on your answers, it will match the features, your characteristics, it will match them to the Games of Thrones characters. That's exactly how we do this. Only instead of Game of Thrones characters, we use genes and proteins and compounds, and so we use biological information. But that's exactly the same principle. So we try to look for example, drugs that have similar features to those associated with longevity. I'll show you a couple of examples that may be easier. But before that, one of the things that's quite incredible in the field of aging is this increase in the amount of data. So I alluded to this already. From our GeneAge database, we have a lot of genes associated with longevity, over 2,000 genes associated with longevity, although it's kind of, as you can see, it's kind of plateauing in recent years. Even more impressive is number of drugs associated with longevity. So we have another database called the DrugAge database of aging-related drugs. So these are compounds and drugs that when you feed them or inject them or give them to animals, they significantly increase lifespan. And we now know over 1,000 of this, and it's increasing exponentially. So there's a lot of interest in identifying these longevity drugs. So we have this database called the DrugAge database. It has over 3,000 entries from 30 model organisms and over 1,000 drugs or compounds that significantly extend lifespan in at least one model organism. So it's really this longevity pharmacology field, as I call it, it's been exploding. It's massive. And not surprisingly, there's a lot of big companies. We were talking about it early on. So a lot of big companies in the space now. One of the first ones was Calico from Google, which was on the cover of Time magazine. More recently, there's Altos here in the UK, in Cambridge, and in the US as well. So there's a number of big players now focusing on one hand because of the advances in the genetics of aging, in longevity pharmacology, and also because there is a huge market to it. I mean, there is at the moment very little we can do about aging. And as I said before, if we could retard aging, even if slightly, to have use health benefits, but also economic benefits. So there's a big increase in the anti-aging biotech sector. But let me go back to the DrugAge database. So there's a couple of things just wanted to quickly point out is that, okay, well, there's a lot of longevity drugs, field is increasing, but of course, some of the drugs are more studied than others, like resveratrol, which you might have heard, and rapamycin, that are much more studied than others. But in terms of effect sizes, it also varies quite a little bit. So resveratrol, even though it has the most studies, if you look at the lifespan changes that you see, it's actually about zero. What that means is that there's several studies showing increases in lifespan from resveratrol, but there's quite a few also showing decreases in lifespan. And when you put it all together, it's pretty much zero. It doesn't seem to have an effect. While rapamycin seems to be the most promising drug in terms of extending lifespan across different systems and different experiments. It's not a massive change, as you can see. So I think in mice is about 10 to 15%. I know it varies between males and females, but I always forget which one it extends lifespan more. But it's about 10 to 15% increases in lifespan. Rapamycin, for those who are not familiar, it's an immunosuppressant drug. It's also used as an antifungal agent. And is used as an anti-cancer agent. It reduces cell proliferation. And it's already used for treating cancer and as an immunosuppressant in humans. And so in mice and other model systems, it extends lifespan about 10-15%. So it's one of the most promising longevity drugs currently that we know of. So one of the things I would say as well is that in terms of the focus of the longevity pharmacology, when you look at all of the drugs that are being studied, that are being developed in the context of longevity, they tend to fall into common themes. So there's a lot of interest in oxidative stress and mitochondria. There's also a lot of interest in senescence cells and senolytics. So senescent cells, I think, as are previously mentioned, so these are cells that stop dividing and then contribute also to inflammation. And there's also quite a lot of interest in mediators of metabolism. Rapamycin would fall into that category, like mTOR, which is target to rapamycin, NAD, and so on. So these are the major focuses of longevity pharmacology, which I actually think is quite narrow. I mean, I think most of the companies working in the space, they tend to focus on quite similar things, and essentially it's the same things. Okay, there's a few variants, but it's the same few options that they tend to focus. I think there's quite a narrow focus in anti-aging biotech, which I think is a problem because we don't know why we age. So, I don't think oxidative damage is a driver of aging. For instance, I go into more details in my Nature Genetics recent paper and other papers I published in the past. I don't think it's important. I could be wrong, but that's what the evidence tells me so far. So there are, I think, a lot of companies focusing on dead ends, in my opinion. And so now, obviously, in biology, you don't know what's going to work or don't. It's always a shot in the dark, but I think it would be nice for the anti-aging biotech field to have a broader focus than it does now. So that's just my interpretation. Now, going back to longevity pharmacology, however, one of the aspects that I'm sure you're aware of that is a big problem is that only a very small percentage of drug candidates actually gets regulatory approval. So it's very difficult to go through. I mean, sometimes there's side effects of a drug. It looks amazing in vitro and in vivo, but then when you go to clinical trials, it doesn't work or it has side effects. So only a very small percentage of the drug candidates actually make it to the clinic. And there's huge costs of clinical trials. So there's a lot of problems. And this is particularly relevant in aging because, at the moment, it's not possible to do a clinical trial for aging. What most companies do is they pick a particular readout, usually disease, and then a disease indication, and then they do a clinical trial for it. That's how most companies do it. Near Bartley in New York is trying to do a first clinical trial for aging, but as far as I'm aware, there's not going yet. So in aging, we have a particular problem. So how to overcome it? So one of the techniques or one of the methods we've been focusing is employing in silico approaches and computational methods to better prioritize targets, better prioritize genes, better prioritize drugs for interventions. So as I said before, I'm not going to go into a lot of details. I'll briefly mention a couple of recent works we've done. The point is to apply network approaches, data systems biology, machine learning, AI for prioritizing candidates. To select those that give you the highest chance of essentially being effective in clinical trials. So to give one quick example, we did this analysis of genes associated with cell senescence. So these are senescent cells. The photos I actually took during my PhD, which I did working in the lab. So we would do a network analysis of genes associated with cell senescence, and that allows us to identify which are the most likely regulators of cell senescence, and allows us to then, we actually did some experiments in cells and identify some new genes associated with cell senescence. And then we can also look at these genes in human tissues and see which tissues with age have stronger evidence of an accumulation of senescent cells, for example, which can help you prioritize and define clinical readouts and outputs. So, this is something we published a few years ago, focused on cell senescence and this systems biology, network biology analysis of cell senescence, going from computational analysis, in vitro studies, and in vivo data. More recently, we also did this work with collaborators in University of Kent Alex Freitas and colleagues, where we essentially looked at longevity drugs from our drug age database. And then from those, we try to look at their features, the chemical features, but also their biological features. I mean, which genes they target, which pathways they target, and so on. And based on those features, then we can predict new longevity drugs. So we go, like I told you, for Game of Thrones, same principle. We look at the features for the longevity drugs, and then we'd go and try to look for other drugs, not yet tested in the concepts of longevity, that share similar features. So you can do that with machine learning and obtain predictions of the most promising longevity drugs. And from one of these analyses, we've done this project, which took several years in C. elegans, where we identified a new longevity drug called rilmenidine. Now, rilmenidine is an oral anti-impartessive drug, so it's used to treat hypertension. It's not approved in the UK. It can be used in some like exceptional circumstances. It's approved in countries like France, so it is used in a clinic. And basically, from our computational analysis, we identified it as a promising longevity drug. And what we showed in C. elegans, so we showed in an animal model, is that it extends lifespan. So that's what you see here. So you see in black, you see the controls, and then you see a dosage-dependent effect. And about 200 micromolar is about the sweet spot. Then after that, it doesn't extend lifespan all that much. And whether you feed it to animals early in life or late in life, it always extends lifespan. So this is the days of animals are alive, and this is the percentage. So you see controls, and then when you feed the animals rilmenidine, it extends lifespan. So we identified rilmenidine as a new longevity drug, and given that it's already used in the clinic, we're trying to do a clinical. Well, we don't have the funding, but we're trying to get the funding to do a small clinical trial to explore it further. We have done a little bit of work on mice, actually, with our collaborators in Harvard, Vadim Gladyshev, where they showed that if you look at, so basically, if you feed mice rilmenidine, then they show changes at the gene expression level that resemble other life-extending interventions. So that's kind of promising in terms of suggesting that rilmenidine may extend lifespan in mice as well. And as I said, it's a promising new longevity drug, a promising new geroprotector that we are very interested in exploring further, and happy to discuss collaborations. Now, the other things, switching gears a little bit, the other thing we've done quite a bit is DNA sequencing. So, as I'm sure you're aware, there's been massive decline in the cost of sequencing. I mean, this is an exponential scale. This is Moore's law for computers, and this is cost of sequencing per genome. As you can see, there's been a massive drop. So it's much cheaper and much easier to sequence genomes nowadays than just a few years ago. I mean, that's so forth with me pretending to work with this sequencing machine. They don't actually let me near it. But thanks to machines like this one, we can now sequence the human genome for less than $1,000 quite rapidly. And so we've been using these technologies as well to study the genetics of aging and longevity, although not in humans, in even more fascinating, well, there's longer-lived species than human beings. So one of the aspects that I've always been fascinated, and Alistair mentioned this early on, is that there is a huge variation in longevity across species. So, I mean, I used to have lots of pets when I was a child. If you have a hamster, it will live about 2 years, maybe 3 years if you're very lucky. If you have a dog, maybe you'll live 10 years. If you have a cat, 20 years, and so on. So there's a huge variation in longevity across species. Humans can live over 122 from Jean Calment, and you have species of whale that can live over 100 years. Some have been estimated to even live over 200 years. So there's a huge variation in longevity across species, even in captivity. So it's not lifestyle. A mouse will age 20, 30 times faster than a human being. You can take extremely good care of a mouse. It will still age a lot faster than a human being. I mean, some of the genetic mutants may age a little slower. They will still age a lot faster than a human being. So there has to be a genetic basis for this plasticity of longevity. It has to be seated in the genome. evolution has sorted out in some way. And and I think Alistair mentioned this as well, so I'll comment on it, because we actually have done some work on this in the past, which is it's not to do with the rate of living. So they use this notion that animals that have higher metabolic rates, they're going to age faster. I think this dates from the industrial age. If you have two machines that are the same and one is running faster than the other, it's going to break down on average, it's going to break down faster. So there was this idea that a mouse ages faster because it's got a higher metabolic rate than human beings. That's not true, because as Alistair mentioned, you have species like bats that have extremely high metabolism, and they can live nearly 40 years. So so it's not to do with metabolic rates, and we've done some analysis on this, which I'm not showing today, but we've actually done a proper statistical analysis of metabolic rates and longevity in mammals, and they don't correlate. So it's not to do with metabolism, but it has to be a genetic basis to it. And so I've been trying to figure it out, and we've done quite a bit of work, which I'll very briefly summarize. It's a little different from what I've mentioned so far. So we've been focusing on a couple of different species. One of them is the naked mole rat. This is actually a photo from so there's a lab in London that has some of these animals, Chris Folks. So these are relatively small. They're a little bigger than a mouse, smaller than a rat. So they're rodents. But while mice and rats can live 2 or 3 years, exceptional genetically modified mice may live 4 years, but generally they live 2 or 3 years. These animals can live over 30 years. And they're sort of the longest lived rodent, even though they're quite small, and they're extremely cancer resistant. So we sequenced the genome in collaboration with some colleagues in Boston and New York. So we sequenced the genome some years back, and we analyzed it. The other species that we focus, I think, is even more incredible, which is the bowhead whale. So the bowhead whale is the second biggest animal on Earth, the blue whale famously being the number one. But they've been estimated to live over 200 years. Now, how they do it? Well, this is indirect methods. I mean, they basically used a biochemical assay of racemization ratios in the eye lenses to estimate the age of whales, of dead whales. Some of them have been estimated to live over 200 years. I mean, there's quite a lot of error margin there. I mean, it's like 50 years or 60 years error margin. I think the point is that still, even if that may be a bit off, the point is that these animals live longer. And I'm convinced that there's other anecdotal evidence that they live longer than human beings in the wild, without doctors or hospitals or vaccines or anything. They live longer than human beings. And if you think about cancer as one rogue cell that just proliferates uncontrollably, then these animals they have 1,000 times more cells than human beings. The only way for them to live so long is because they have anti-tumor mechanisms that we lack. I mean, that's the only explanation. So they must have evolved mechanisms to suppress tumors that humans don't have. So we sequenced the genome of the bowhead whale. I won't go into details. We identified several interesting candidates, so like genes that in the bowhead whale are slightly different than other mammals. Like, for example, genes associated with cancer, cell cycle proliferation, histone deacetylase is involved in epigenetic regulation, ERCC1. You might remember that I mentioned a mouse mutant of ERCC1 early one. So if you mutate ERCC1 in mice, they age faster. So bowhead whales have only a few very specific changes in ERCC1, which we can actually predict using protein models, that you see here, that affect the interaction with other proteins associated with cell division, DNA damage responses, and they've also associated with cancer in humans. So, there's a number of interesting gene candidates. Obviously, all of this is computational because we can't really go and get a bowhead whale and study it in the lab. So what I would like to do if I had the funding was to actually take some of the genes from the bowhead whale or some of these mutations and make a mouse with these mutations and see if they're cancer resistant and if they live longer. That's how we'd like to do. The point is that there's a lot of interesting biology and potential genes protecting against aging and diseases in these long-lived species. That's the point. We make all the data available, from the naked mole, the bowhead whale. Everything's available online. So the point is that biomedical research mostly focuses on disease-prone organisms, like mice. Like mice develop cancer a lot, develop diseases a lot. So this is a complementary synergistic models. Naked mole rats and bowhead whales, they're disease-resistant organisms that may help us identify genes, processes, pathways that protect against disease and protect against aging. So I see them as complementary in biomedical research. And so in the last few minutes, I'll just come back to why we age. As I said before, we don't know exactly why we age. What's the molecular drivers of aging? The number of hypotheses, oxidative damage, telomeres, DNA damage. I think there is some evidence for, if you look at DNA damage, for instance, if you disrupt DNA damage in mice, it does appear to accelerate, in some cases, aging. So there is some evidence in that regard, but we haven't been able, for example, to create mice that live a very long time by having better DNA repair mechanisms. So there's still a lot of unknowns in the field regarding what are the drivers of aging. But I wanted to put another idea in this context, something we've been doing some work, which is, and it derives from some of our work on comparative biology. So this is, what you see here is a correlation between longevity and developmental timing. So here you have, this is log scales, and here you have, x-axis age of sexual maturity, how long it takes animals to reach sexual maturity, and here you have longevity. Actually, it's adult longevity, which is how long animals live minus age of sexual maturity. And what you see is a very strong correlation. It's extremely strong across mammals. So each of these dots is a mammal. And so what that means is that how long for a species, not for an individual, but for a species, how long animals of a species take to reach maturity, how long it takes them to develop, is an extremely strong predictor of how long they live afterwards. That's the point. So one hypothesis I put forward last year, which I call the software design flaw hypothesis, is that, what if aging is not a result of molecular damage? It's not damage. It's not DNA damage, oxidative damage, it's not damage. Instead, it's just a product of the running of the genome and the epigenome. So, it's, let me elaborate on that a little bit. So the idea is that, aging is not damage. It's not molecular damage that builds up. So that's the traditional view. Aging is the damage that builds up, DNA damage, oxidative damage, telomere damage just builds up and becomes too much and you die. What if it's not that? What if it's actually regulated process that runs in the genome and epigenome? So we know, and this is the non-controversial part of my hypothesis, we know that development can be seen as a software that runs in the epigenome. So we have the DNA sequencing, which you could see as a software program, a highly complicated, very complicated software. And then it runs in the epigenome. It runs in methylation and chromatin modifications and other, because the genome is the same across cell types, but it runs in the epigenome that indicates differences between cell types and developmental stages. So as we develop, you're going to have changes in the epigenome. I'm showing methylation here, but that's broadly defined. You see changes in the epigenome that result in different programs running in different cells and at different times, right? So I don't think that's controversial. That seeing John Maynard Smith, for instance, used similar analogies of seeing developments as a software program that's encoded in the genome and runs in the epigenome. What's more controversial, and that's the hypothesis, is that, well, what if this impacts on aging? So the hypothesis is very simple. I think it's quite miraculous that, we start as a single cell. Each of us started as a single cell. And I think that's quite incredible. If oxidative damage or, DNA damage was such a big, huge, force that was, we couldn't get out of, then, how do we develop in the first place? That clearly we undergo huge amount of cell divisions, we grow and we develop early in life. And that's fine. We become functioning adults. So the hypothesis is that, well, there's a number of programs that runs in our cell during development and growth. But then the hypothesis is that as a form of what's called antagonistic pleiotropy, perhaps some of this routines, some of these programs in development, they just continue later in life and they become detrimental. So that's the hypothesis. And this runs in the epigenome, results in gradual changes in the epigenome. I mean, there's a couple of classical examples of this. One of them is presbyopia, which is short-sightedness. So basically your eye lenses, they grow continuously throughout life. So when you reach about 40, and I've noticed this because I'm over 40 now, if I want to read something, if I want to read a book, I have to take off my glasses to read up close. And that's because it just keeps growing. Your eye lenses just keep growing and they become too thick, and then it can't focus properly. So that's a classical example. Thymus involution would be another example. The thymus involutes from very early in life, about once you're 1 year of age, all the way through life. So, these are good examples of processes that start very early in life. They're optimized for reproduction, but then continue late in life and become detrimental. And the idea is that this running is reflected in cells as they change epigenetic states from development to later on to old age, and then it becomes detrimental. So the hypothesis is that, there are definitely some aging phenotypes and diseases that are due to damage. Cancer would be an example. Cancer is due to damage. It's due to mutations primarily. There's other factors associated with aging. That's a different talk in itself. But the point is that definitely some damage does play a role in some aging phenotypes like cancer. But my hypothesis then is that aging is an information problem. And what we need to do is decode our genetic software and epigenetic information and how it flows in space and time in our bodies in order to understand the aging process. So that's the hypothesis. And as I said, I published about a perspective on it last year in Genome Biology, if you're interested in more details. And so in summary, and running out of time, so aging remains a mystery of biology. We don't know why we age. There's a number of hypotheses: telomeres, oxidative damage, DNA damage, epigenetics, and so on. In humans, none of them have very strong evidence, none. So, I'd be very skeptic about anyone who claims to know why human beings age. We don't. So aging remains a mystery of biology, but there has been advances. We know of over 2,000 genes that can modulate aging in mortal systems. That's quite remarkable. I think it's a huge advance. I think you should get the Nobel Prize. Tom Johnson, Cynthia Kenyon, these early pioneers. I think you should get the Nobel Prize for it. It really revolutionized our thinking about aging. It means that we can manipulate aging by single gene manipulations and drugs as well, as I mentioned. I hope I've persuaded you that we can use computational methods to identify new genes, new processes, new drugs associated with aging and other processes as well. One new compound we discovered from using these methods is rilmendin, which we discovered as a new geroprotector, a new longevity drug, already approved for clinical use, and so we're now trying to repurpose it and do a small clinical trial. I told you about our studies of long-lived species and species differences in aging and trying to find genes that protect against aging, which is complementary / synergistic with existing biomedical approaches. And lastly, I told you about the hypothesis that perhaps aging is not damage, but rather it's an information problem, which is something I'm quite interested in exploring. And so with that, let me just we're based at the University of Birmingham here in the Queen Elizabeth Hospital, and we have a brilliant group of talented and ambitious students and postdocs. So they're really the ones who've done the work, and of course these are the funders that paid for the work that I told you about. If you're interested in any preprints or PDFs, they will all be available in our lab website, or feel free to drop me an email. And yes, happy to thank you very much for your time and attention. Happy to take any questions.